So good day everyone, welcome back to Magic School. So today we'll be going into absorption and transport of amino acid. This is under our metabolic pathway course, and we're talking about how amino acids are actually being absorbed in the body. Yeah, so let's get into it. Now, amino acids are gotten down after the breakdown of proteins. So now in the intestinal cells, these amino acids are transported from the intestinal lumen into the intestinal mucosa cells, into the cytosol of the intestinal mucosa cells. And from the intestinal mucosa cells, they're then transported into the portal circulation, and then they get to the liver where they are metabolized. So now there are two mechanisms by which this amino acid gets into the cytosol of the intestinal mucosa cells. Now the first one is the carrier protein transport system, and the next one is the glutathione transport system, which is the same thing as gamma glutamate cycle. So now this carrier transport system uses carrier proteins present in the plasma membrane, present in the membrane of these cells. It transports these amino acids into the cell. There are also different carrier proteins for different groups of amino acids that transport these amino acids into the cell, such as neutral amino acids, basic amino acids, acidic amino acids, and some other amino acids. So yeah, there are actually 7 of them, 7 carrier proteins for this. And now this process uses just one ATP, and then it is the most important and the main system of absorption for amino acid in the body. Now, but let's actually look at it, look at an image so we can understand it better. So from the diagram, we see where is labeled the intestinal lumen, and then the diagram there with the brush border is the intestinal mucosa cell. So now the amino acid is being transported into the intestinal mucosa cells with sodium. So now this is a symporter that transports amino acids and sodium into the cell. Now when they both get into the cell, the sodium is transported out of the cell in exchange for calcium ion, and in this process here uses ATP. That transporter that is there is also called the sodium pump. It uses ATP in the taking out of sodium out of the cell and bringing out of calcium and bringing calcium into the cell. Now the amino acid that was brought into the cell is then transported by another transporter from the intestinal mucosa cell into the portal vein, and that's how we get into the portal circulation and get to the liver. Yeah. So now the next mechanism of the absorption of this amino acid is the glutathione transport system, which I also said is the same thing as gamma glutamate cycle. Glutathione is actually a tripeptide composed or comprising of glutamic acid, cysteine and glycine. Now this glutathione can also be written as GSH, the G there signifying the glutathione, while the SH there signifying a sulfhydryl group. Now this sulfhydryl group is from the cysteine that is present in the tripeptide. So now in this mechanism, one amino acid being transported requires 3 ATP, while in carrier protein transport system just 1 ATP is being used for just one amino acid. So actually the first one, carrier protein transport system, is better or is preferred by the body system because it uses less energy, and that way is able to save energy for other processes. So now let's look at the diagram of this gamma glutamate cycle. So now in the beginning, the GSH, which is the glutathione, is acted upon by gamma glutamate transpeptidase, and then it cleaves glutamate from the cysteine glycine, and then this glutamate is conjugated with amino acid, and then it's transported through a transporter into the membrane, or through the membrane into the cell, and then gamma glutamate amino acid is formed, and then the cysteine glycine is left on its own. Now this gamma glutamate amino acid is acted upon by another enzyme, an amino acid is released, and 5-oxoproline is formed. Now this 5-oxoproline is acted upon by an enzyme that uses ATP to form glutamate back. Now glutamate reacts with cysteine, the cysteine and glycine that was brought in cysteine glycine is then broken down to glycine and cysteine. So now the glutamate reacts with the cysteine to form gamma glutamyl cysteine using an enzyme also that also requires ATP. So now that is actually 2 ATP that has been used. Now this gamma glutamyl cysteine reacts with the glycine to form the glutathione again using another enzyme, and this enzyme also requires ATP. So when we look at this entire cycle here, we see that 3 ATPs are actually being used just for the transport of one amino acid inside. Now the glutathione that is formed again is then used to transport another amino acid inside, and the process goes on. So now this is also an image that depicts what I just explained earlier, but shows a peptidase that actually helps in cleaving the cysteine and glycine to form cysteine and glycine. Now we're talking about absorption of amino acid in different tissues in different parts of the body. The extrahepatic tissues, the muscle cells, and then the hepatocytes, which are the liver cells. So now amino acids can be found in different parts of the body. So now in different parts of the body, these amino acids can then be broken down, or we then get ammonia from these amino acids. And now this ammonia needs to be transported down to the liver so that they can be excreted from the body or used in biosynthetic pathways. So now this is where the formation of glutamine comes in, using glutamyl synthetase. So now the formation of this glutamyl occurs in two steps. The first step is the formation of the gamma glutamyl phosphate from glutamate, and then the next is the formation of glutamine from this gamma glutamyl phosphate. Now this glutamyl is a non-toxic form of ammonia. It can be called that is used to transport ammonia in the blood. Ammonia is quite toxic in the body system, so it has to be regulated. So now it is converted to glutamyl, or attached to glutamate to form glutamyl, and then transported in the blood to get to the liver and into the mitochondria, and then for it to be metabolized and broken down back to release ammonia, which can then go into the urea cycle. Now let's look at metabolism of amino acids in muscle cells. In muscle cells, when there's a need for energy, amino acids can be broken down or degraded to release energy. So here, these amino acids are transaminated pyruvates to form alanine, and then this alanine is transported from the muscle cells to the liver cells, where it is finally metabolized. Not just any amino acids metabolized in the muscle cells. Part of them are the branch chain amino acids, such as the leucine, the isoleucine, and valine. They are metabolized in the muscle cells for energy. So now lastly, in the liver cells, which can also be referred to as the hepatocytes. Now amino acids present in the liver cells are transaminated to form glutamate from alpha ketoglutarate, and then those amino acids that were actually transaminated, or amino groups were removed, then form their respective alpha keto acids. And then now this glutamate that is being formed is then taken into the mitochondria of the liver cells, where it undergoes deamination by glutamate dehydrogenase. So now this enzyme converts glutamate to alpha ketoglutarate, donating its amino group, the amino group of the glutamate, to urea cycle to form urea, or to biosynthetic pathways that actually needs it. And then glutamine that was being transported in the blood, or that was formed in extrahepatic cells, and then are being transported back to the liver, get into the mitochondria also, and then they're acted upon by the enzyme called glutaminase, and then this converts glutamine back to glutamate and the amino group. Now the amino group also can enter the urea cycle. So this entire process that we just talked about is in the liver cells can also be referred to as transdeamination. A transdeamination reaction is a reaction that involves an aminotransferase and involves the glutamate dehydrogenase. So most amino acids that are metabolized undergo this transdeamination reaction that involve the aminotransferase and then the glutamate dehydrogenase. But now some amino acids, they don't undergo this transdeamination reaction, but these amino acids, they bypass transdeamination. So an example we have is proline, we have lysine, we have threonine, and then we have hydroxyproline. And you can also check out our video on urea cycle to be able to understand how this ammonia that is being released is used up, or is used in the urea cycle to produce urea. So that is that for this video. So don't forget to like and subscribe. Thank you.